Hi friends, today we are going to see the past year NEET questions for the chapter Cell, the Unit of Life. And today in this video we will cover part 4. Now let's begin with the first question here. Which one of the following structures is an organelle within an organelle? Okay, so this question was asked in the year 2012. So what does this question state? It is asking us an organelle which is present within an organelle within an organelle, right? Okay, so you're given 4 options here: endoplasmic reticulum, peroxisome, mesosome, rhizosome. Yes, the correct answer is option A, ribosome. See, as we already discussed a lot about ribosome, this ribosome are small, they're naked, that is they don't have a membrane bound or a membrane around them, right? They are particles which are made up of rRNA and proteins. They are made up of rRNA plus proteins, right? Ribosomes are also seen in organelles like mitochondria and chloroplasts. They are the cells of protein factories, and they are found on the rough endoplasmic reticulum, and also they are scattered inside the cytoplasm as polyribosomes. Therefore this ribosome, since it is amembrane, that is it does not have any membrane around them, they are found within organelles scattered, or it might be present on the surface of rough endoplasmic reticulum. Therefore the correct answer to this question is option A, ribosome. Okay, in the solution part they have explained the same thing what I said now. Right, now let's move to the next question here. This question was asked in the year 2012. Which one of the following cellular parts is correctly described? Right, so now go to the solution. Let's go one option by option. Centrioles, site for active RNA synthesis. Lysosomes, they have optimally active at a pH of 8.5. Thylakoids, they are flattened membrane sacs that are present in the grana of chloroplast. Ribosomes, those on the chloroplast are larger than 80S, while in the cytoplasm are 70S. Of course option D is wrong, we can understand it from that. So which is the right answer now? The correct answer is option C, thylakoids. Why? We already saw in the earlier parts, they are flattened surface or sacs which form or which is present in chloroplast. But here they have correctly given you what is thylakoid here. They are actually flattened membrane sacs that are forming the grana of the chloroplast. Okay, and if you see the first one, centrioles, they actually serve as basal bodies for cilia and flagella. So the right answer is not that. Lysosomes, if you see, they are membrane bound organelles, and they are not supposed to be active at 8.5 but an active pH of 5. Okay, they are active around a pH which is quite acidic in nature. So therefore option B also becomes wrong. Option D, it is very well understood it is wrong, because we already discussed about the prokaryotic and the eukaryotic ribosome presence, 70 and 80S. Therefore the correct answer is option C, thylakoids. Okay, now let's move to the next question here. Now this question was again asked in the year 2011. Important site for the formation of glycoproteins and glycolipids dash. Okay, now what is glycoprotein and glycolipid? They all have glyco in it. One is protein and one is lipid. Now when I say glycoprotein, they are any class of protein which have carbohydrate group attached to the polypeptide chain. And when I say glycolipid, they are lipids attached with carbohydrates. So you have protein attached with carbohydrate, and here you have lipid attached with carbohydrate. So that is the difference between glycoprotein and glycolipid. And if you see these two things, what is the function? If you see, they are responsible to maintain the stability of the cell membrane. Very important function, maintaining the stability of the cell membrane, and it actually facilitates or it helps in the cellular recognition. And this important site, if you see, the correct answer goes to Golgi apparatus. The correct answer is option B. Okay, now to your right side you can see how the Golgi apparatus will look like. right? So what is the function of glycoprotein and glycolipid? They help in cellular recognition and maintains the stability of the cell membrane. Now next question. I'm sorry, peptide synthesis inside a cell takes place in dash. Again, 2011. When I say peptide, they're talking about protein, right? Okay. So here we've already discussed this question or related to this question a lot in the earlier parts. So therefore the correct answer to this question is ribosome. We already saw this ribosome has rRNA plus protein, right? So protein, peptide, they mean the same. Therefore peptide synthesis inside the cell takes place in the ribosome. The correct answer is option D, ribosome. Now here if you see an interesting feature about ribosome, a ribosome will have a larger and a smaller unit. So that is something unique about ribosome. It will divide itself into two parts. One will be smaller unit and one will be the larger unit. Okay, so can you see the difference here? They come like a finger-like projection here. Okay, now let's move to the next question here. This question was asked in the year 2011. What are those structures that appear as beads on the string in the chromosomes when viewed under the electron microscope? So yeah, this is also an easy question. See, they're asking us, when you view it under the electron microscope, they will observe, or they will be observed as strings. Okay, now genes, no. Base pairs, no. Okay, it should either be nucleotides or nucleosomes. Nucleotides, the structure is quite big when compared to nucleosome. Therefore the correct answer is option C, nucleosomes. Under electron microscope, they appear as beads on the string of the chromosome. Why? If you see, as I already said, nucleotide is quite larger or longer, so this is shorter. Okay, so therefore they have that appearance, shorter length of DNA wrapped around a core of histone proteins. So this is the answer for this question. Now let's move to the next question here. See, this question is asking us to actually figure out what the picture is and the parts. Okay, so what is this picture actually? The figure below shows the structure of mitochondria. Yes, it has 4 parts. Now you have to tell me each part what it is. Okay, select the part correctly matched with its function. So you have part D, part B, part C and part A. Now look at part D. Okay, now look at part D. They have said it's the outer membrane which gives rise to the inner membrane. And B, they have said it's the inner membrane which forms the cristae. A, they have said it's the matrix, and C, they have said it's the cristae. So the correct answer is option B. Inner membrane which forms the infoldings called as cristae. So this part is clearly, or it is correctly matched for its function. Okay, so the mitochondria, as you all know, it has cristae, all that is there. But part B is correctly matched. The mitochondrial inner membrane forms inner foldings called as cristae, which allows greater surface area. Imagine this cristae is not folded, then mitochondria will actually occupy a whole space in your body. So that is why God created this cristae, so that surface area has been consumed in a lesser amount. Therefore the correct answer is option B, part B. Now let's move to the next question here. This question was asked in the year 2011. In mitochondria, proteins accumulate in dash. Okay, so in mitochondria, where will the proteins get accumulated? Any idea? The correct answer goes to option B, inner membrane. We already saw that mitochondria, the inner membrane, in the earlier slide we saw the inner membrane will give rise to what? Infoldings. And what are the infoldings called as? The infoldings are called as cristae, right? And this will actually allows a less consumption of the surface area. So these things we saw, less consumption of the surface area. Okay, So this inner membrane of the mitochondria contains more than about 151 types of or different polypeptides, and here if you see, they have a very high protein to phospholipid ratio. So the proteins, where do they get accumulated? In the inner membrane of the mitochondria. Okay, option B is the right answer. Now let's move to the next question here. This question was asked in the year 2011. Can we read it? Which one of the following is not considered as a part of endomembrane system? Endo means inside, right? It is present inner or inside. Exo means outside. So they are asking us the one which is a part of an endomembrane system. Golgi complex is not the answer. Lysosome is not the answer. So it should be peroxisome, or it should be vacuole. The correct answer goes to peroxisome. So what is this peroxisome? It contains enzymes which will, so what does it contain? It contains enzymes. Okay, it will contain enzymes that will oxidize certain molecules that is normally found inside the cell, especially fatty acids. Okay, fatty acids, then it can be amino acids. Okay, so what actually happens here is those oxidation processes that happens here, the reactions that happens here will produce something called as hydrogen peroxide. What does it produce? Hydrogen peroxide. Okay, hence the name, I'm sorry, hydrogen peroxide. So hence the name peroxisome here. Okay, see, it will contain enzymes and this will oxidize these kind of molecules that enter into them, and this oxidation will lead to the release of hydrogen peroxide. That is why it got its name as peroxisome. So here if you see, this is the endomembrane system here. Out of the 4 given here, the correct answer is option B, peroxisome. Now let's move to the next question here. This question was asked in the year 2010. Right, can we read it? Which one of the following structures, okay, which one of the following structures between two adjacent cells is an effective transport pathway? Right, they are talking about the transport pathway. So what do you mean by transport pathway? We have two different pathways. One is active and the other is passive. So here they are asking us the structures between two adjacent cells is an effective transport pathway. Okay, we are given 4 options here: plasma lemma, plastoquinons, plasmodesmata, then you have endoplasmic reticulum. Of course it is not endoplasmic reticulum. The correct answer is option A, plasmodesmata. Okay, they are the structures which are present between two adjacent cells, which actually permit the transport and communication between them. Okay, so this particular feature or this particular structure called as plasmodesmata, they are responsible for the transport and communication which happens between the two adjacent cells. They are the fine cytoplasmic strands that connect the protoplasm of the adjacent cells. Thereby it will pass through the cell walls. Okay, plastoquinons, endoplasmic reticulum, plasma lemma, they do not have any transport system. They are all associated with the cell structure, but they are not associated with any transport pathway. If at all they are involved in the pathway, they will not be effective as how a plasmodesmata is as effective. Okay, therefore the correct answer is option A. Now let's move to the next question here. Now this question was asked in the year 2010. Now let's read it here. Which one of the following has its own DNA? I think this is an easy question for you to identify. Can anybody guess? Out of the 4 here, lysosome, no, it doesn't have its own DNA. And peroxisome, if you see, it's an endomembrane organelle, and of course that too does not have a DNA of its own. Therefore the correct answer to this question is mitochondria. Why? Can anybody guess? See, mitochondria has its own DNA and it is a structure. Okay, it is a structure within the cytoplasm. We all know that, right? It is a structure within cytoplasm of eukaryotic cells. And what does it do? What does this mitochondria do? It actually carries out aerobic respiration. What do you mean by aerobic? Aerobic, there are two types of respiration, aerobic and anaerobic. Anaerobic is without the presence of oxygen, and aerobic is with the presence of oxygen. So here, if you see, it undergoes aerobic respiration, and this mitochondria will act as a site for TCA cycle or Krebs cycle. Okay, therefore it is very important, since we have the energy coming out from this particular organelle. So this organelle, if you see, it is very special. It produces ATP, and that is the source for the whole body to survive and to have energy. Therefore this mitochondria is blessed with its own DNA. Now you understand the correct answer now. The correct answer is option A, mitochondria. So whatever I explained here is what they have given in the solution part also. So I've clearly told you why a mitochondria has its own DNA. Now next question here. Okay, this question was asked in the year 2010. The main arena of various types of activities of the cell is dash. Main arena, the main organelle or the main part where the activities of the cell will take in charge with a main or a booster, or you can tell it's the main site actually. Okay, so you're given four options here: plasma membrane, mitochondrion, cytoplasm, nucleus. All four parts seem to be important for you, but out of the four, only one stands to be very important when it comes to various activities. So if you look at the four options given here, each one has its own activity, but when we say various, it means many, right? So there is only one organelle which is responsible for many activities. Therefore the correct answer goes to cytoplasm, because it forms the living protoplasm of a cell excluding the nucleus, and it will consist of proteins, it consists of lipids, it will have carbohydrates, many more molecules in them. So here they have clearly mentioned various types of activities. If they haven't mentioned this, then you would have got confused of which one is actually, because all the four options given here are main. But when they talk about various activities excluding the others, then therefore the correct answer is option C, cytoplasm. So already, as I said, they have proteins, fats, nucleic acid, vitamins, and many more associated with it. Okay, now let's move to the next question here. Now this question was asked in the year 2010, again in the year 2010. Now let's read this here. The plasma membrane consists mainly of dash. Okay, we all know the plasma membrane is very important membrane of the cell, and it forms and it actually contributes in a lot of activities, and it helps in the cellular functions. Now this plasma membrane, if you see, it comprises certain things, or it has certain molecules embedded on them which actually are in dominance. Okay, you have certain molecules which have a dominant embedding over their surface. So any idea what it could be? See, plasma membrane, if you see, they are comprise of proteins. Mainly it will be protein. Okay, and this particular protein, if you see, will be embedded. As I already said, it consists mainly of proteins, and this protein will be embedded in the phospholipid bilayer, and protein molecules occur at places both inside and outside the lipid bilayer. Therefore the correct answer to this question is option B, proteins embedded in the phospholipid bilayer. So now you can see the diagram of the plasma membrane. It has the protein, the alpha helix protein there, the outer membrane, the hydrophobic region. What do you mean by hydrophobic region? That is here. Can you see it here? Hydrophobic region means where there is no water requirement. Okay, it will not allow the entry of water. right? So this is how a plasma membrane appearance will be in a 3D structure. Now let's move to the next question here. Now this question was asked in the year 2009. I think this question is very easy for you to answer because we already discussed what is a plasmodesmata in the earlier slides. Plasmodesmata are dash. So what is their activity, or what is their function? See, plasmodesmata are the connections between adjacent cells. That is what we saw in the earlier slides. We talked about the adjacent part, right? So here if you see, they are the connections between the adjacent cells. Plasmodesmata are the narrow channels that act as intracellular cytoplasmic bridges. So what was the function that we saw of plasmodesmata? They help in the communication process. Important function is a communication process. Okay, so here they act as cytoplasmic bridges. They facilitate communication and transportation. So what do they transport here? They transport materials, and this plasmodesmata is very active in plant cells when compared to eukaryotes. Plasmodesmata are formed during cell divisions, and what happens after that? If you see, in trace amount you will find them in the endoplasmic reticulum. Okay, the correct answer goes to option C. They are the connections between the adjacent cells. So as I already said, this has many functions. One is communication, the other function is transportation. Okay, transportation. What else we saw? Transportation of different materials. Okay, during cell division it will be in a prior amount or in dominant amount, but later on it will be found in traces in the endoplasmic reticulum. Okay, and later on again when it undergoes cell division, this will be in prior amount. Okay, therefore the correct answer is option C. Fine, now let's move to the next question here. This question was asked in the year 2009. Cytoskeleton is made up of dash. I think by now you've guessed the answer because we already discussed this in the cytoskeletal part. We talked about microfilaments and microtubulins. When I say microfilaments, they are made up of actin proteins, and microtubulins, they are made up of the protein called as tubulin. By now you have guessed the answer. Therefore the correct answer is proteinaceous filaments. Cytoskeleton is involved with microfilaments. Always remember that in your mind. It is very important for you to understand what a cytoskeleton, the backbone of the cellular organization. So therefore the correct answer is option C, proteinaceous filaments. Proteinaceous filaments are also called as microfilaments, and you have another system which is called as microtubules. They are also a part of cytoskeletal formation, but not in prior amount when compared to microfilaments. So as I already said, microtubulin have tubulin as protein, and microfilaments will have actin as proteins. Therefore the correct answer is option C. Okay, next question. So this is the last question for this part 4. Okay, let's go. Stroma in chloroplast of higher plant contains dash. Right, so this question was asked in the year 2009. So what do you mean by stroma? Have you ever heard what is stroma, or any idea where is it present? It is present in the chloroplast. Okay, it is present in the chloroplast of higher plants. Where are they present? They are present in the chloroplast of higher plants especially. So what do they contain? What does the stroma contain? Stroma contains light. See, it should either contain light dependent or light independent. Okay, stroma, as I already said, they are the chloroplast. They are present in the chloroplast of higher plants, and here if you see, they will be light independent reaction enzymes. Okay, they are the light independent reaction enzymes, and within the stroma are the presence of thycaloids. They are the flattened surfaces that are present in the grana of the chloroplast. We've already studied about it in the earlier slides, right? So these thycaloids, if you see, they are present in the light independent reaction enzyme part, and sometimes they may also have some sub-organelles. They may also have some I'm sorry, they may also have some suborganelles in them. Okay, which are a site of photosynthesis. Therefore, the correct answer to this question is option D, light independent reaction enzymes. So, the correct answer is option D. Okay, so with this we complete part 4. Thank you for watching my video. Hope we will all meet in the next part of the same chapter, Cell, the Unit of Life. Thank you again.